All right, thank you. Thank you for this introduction. Thank you everybody for being here. So I will do a very quick recap of what we did last week and then, get started. So this is live coding. Which I said, this is a little bit different from a standard tutorial. The biggest difference is in a standard tutorial, generally, and I've done many of these, and the instructor kind of cheats in the sense that, we do everything beforehand. If I'm doing a tutorial, I have everything beforehand, and then anything I do live, I'm actually kind of just copying and pasting from previous notes. Whereas here, I have an idea of what I want to do, but I don't have any previous notes for copying and pasting. So we're sort of a little bit making it up as we go along. So for the benefit of refreshing everyone's memories, and also, if someone did not join last week, so this is, Lawrence also posted the link in the chat. So everything is on GitHub. There's also, and the data then is on Dropbox as well. And actually, someone asked during the coding, if you're not familiar with GitHub, so actually everything is also here on Dropbox. And then there's this data folder, and this data folder has I forget, I think it's about 200 of these, or 100 something of these files. Each one of them corresponds to a mag, to the output of, in this case, semibin2, but it could be any mag binning tool or any mag producing tool. And then these happen to be dog gut microbiome samples, but it could have been anything at all, right? And so, what did we do last week? So last week, so we did a couple of things. So first, we used Pixie to install the tools, and then we used install Jug and checkm2, and then we built this very simple script that first downloads the checkm2 database and then runs checkm2 on all of our mags. And I said that we didn't have the time to run it during the live stream because it takes a couple of hours, not too long, but a little bit longer than the stream ran for. So afterwards, I let it run, and then it produces this output. So I have here the output. I have actually not looked at it at all. We will start looking at it today. So this is the output of checkm2. So this is what so it produced a log file. So let's just check one by one what these files are. Obviously, we can guess that this is some sort of log. Okay, we can see, yes, so this was 134 bins. So this was started just at the end of the stream last week, and then took about half an hour, and then finishes at 10:25, finished successfully. Okay. Okay, not particularly informative with respect to the results, but nice to know. Think then some files here, some files there. I think these are internal files that we don't need to worry too much about. And then there's this one file, quality report.tsv. This is where the main results are. So this is what now we will look at this, and we will write a little bit of code with polars and matplotlib, and we will do some summary statistics and maybe a couple of plots just to have maybe a better visualization of our data. So we've been using Pixie. So actually first let's add polars. Is it add polars? Sorry, I did not mean to do that. Sorry, I did not mean to do that. So, okay, so that was a bit silly, but we want to add polars. And we want to add matplotlib. And I think we want to add IPython, so we can run it. Okay. And so again, so the way Pixie works is, even though I'm using this interface where, I'm using an imperative interface where I'm adding on the command line, all this does is it is changing these two files, pixie.2ml and pixie.lock. And together these two files completely define the environment I want. So I don't need, so in particular, it doesn't matter which order I added the packages. It doesn't matter anything except the content of these two files. So let's just test this out. Try Python. Yep, this is and I think I might need to from that. Let me just, okay, so this is working. It shows a little window on my screen. This is what I wanted. So I'm gonna add, okay, so this pixie.log file, generally speaking, I don't want to actually, this is not a human file. This is a file for the computer, and then add polars. Right. Okay. Yeah. Okay, and I'm gonna push to GitHub, right? So now Python import polars as PL. So I think this is the standard abbreviation used for polars. CSV and checkm2 and quality report and separator equals. So this is a tab separated file. Just see. Yep. And so the way I'm going to be doing things here is something I usually do, which is I'm going to be doing most of the coding in an IPython shell, and then I will save some of that to a script if I want to keep it. But the initial exploration, I'll just, run it on an IPython shell, and then, not actually keep it. This is just for our, or in this case for my edification. All right, so you can see. So one thing in this case, I've taken these mags. These are from a project from Anna Kushko, who's a postdoc in my group, and so she had actually already ran through all of this. So and in particular, she had already named them in such a way that the lower numbers correspond to better mags. So we can see that the 000 is, what checkm2 considers a perfect mag, 100% complete, 0% contamination. And as we go down, the last ones are not as great. But let's see if we can. All right, let's see. So actually what I want to do first, okay, US columns. So okay, we have these are the columns that we get. Completeness content. So let's So actually, even before that, let's do this. Before we do any plotting, we can use this describe function, which will give us the percentiles, completeness, and contamination. And okay, this is actually quite good. So the completeness, the median, so that's the 50% percentile, is 98%. And then, you know, so in this case, as I said, these are already actually processed, so that they already were filtered so that the minimum value is 50, but we can see that they are quite good. You know, this would be maybe something, you know, in this case, I trust Anna's work, but you know, if we had gotten the mags from somewhere else, maybe, you know, we would be running them like this. So this is quite nice. And then one thing we can do now is just plot plot completeness against contamination. Okay, this is quite nice. Okay. Oh, yeah. Okay. I actually want seaborne as well. So okay, I'm gonna do this thing. Quality graphs, quality figures. Okay. I also want to add seaborne, so I'm gonna add seaborne. So if you're not familiar with seaborne, so has its, some people really like it, some people like it less. So but seaborne is it builds on matplotlib, so it's basically plays well with matplotlib, and then it adds a bunch of other functions to make several different types of plots. So in this case, actually, I just wanted a fairly standard. So I'm going to go back to the shell. I'm going to add the pixie files again. I'll add seaborne. Okay, and now so this is what I said before, so this is often what I do. So we start with the first version is on the command line, and then we can save it and start editing it to be a little bit nicer. So this is actually what I wanted, was to despine. Yeah, I want to save. So let's save it. Let's make sure we create the output directory. Like there's plot. Okay, so this gonna create the directory plots. If it exists, okay, meaning, you know, no error. And then and then we can create this initial plot. Okay. Here. Okay. Oh, okay. Failed to import any of the following Qt binding modules. Sorry, I'm going to do something. I'm going to make my prompt a little bit smaller. Yes, there. That's better. So normally I use slightly smaller fonts. So this is our very large fonts because I think it works better on stream, but just to be sure. But here, okay, failed to import any of the following Qt binding modules. So I think this is about the, this is a Pixie add. So this is a, yeah, okay. So this is for matplotlib. I think I needed this to be able to plot it. So, okay. I think this might work. Oh, okay. Yeah, that works. Yep, this is the plot. Okay, right, so this is the plot we had before. Okay, again, I'm gonna add by Qt. I'm not sure. I find this, frankly, some of these things are a little bit mysterious to me. Why sometimes you need it, sometimes you don't, but apparently now you do. So apparently you need it to save a plot, but not to have it show up on your screen. I'm not sure exactly what's going on. All right, but this works. Okay. Okay, I'm gonna. All right, let's see if we can do a little bit better. And so actually what I would like to do, what I would like to do, and I'm going to assume you can see my screen here, or what I'm going to add here to the screen. So what I'd like to do is I would like to add to both the X and Y axis some sort of histogram, so that we'd have something like this for the contamination, and then also something like this for the completeness, right? And so that we would be able to see a little bit better. Right, so I'm gonna. So this will now be my. So this will now be the goal for the next, hopefully few minutes, and then we will also look at a little bit at the rest of the data that we have. But let's start there. Let's start to see if we can. So we can generate a nicer picture, or this is a nice picture, but an even more informative picture. So again, we have QS. And so let's start with something a little bit simpler. Which is a histogram. Okay, so this is what the histogram looks like. I'm gonna. So we had seaborn. Wait. Okay, I want the bins to be slightly smaller. Bin width. So what I'm trying to do here. No bin width. What's bin width? Bin. Okay. Yeah, something like this. So this is the completeness histogram. See how it looks for contamination. Okay, yeah, so this is probably not, so for contamination, probably I want something like this. So that the bin width is 0.2, because it only goes between 0 and 10. Again, because these are already pre-filtered by Anna. In real data, you probably might have more. But, okay. So then, so I often say that there's actually two reasons to plot things that are very different. So one is what I'm doing now, which is, it's very good to explore the data because, our brains, generally speaking, are much better at grasping this than this. So if I show this, even if, I can get an idea, it's pretty nice, informative, but it's much faster, I'm much faster at processing information by looking here. So that's one reason plotting is great for data exploration, and this is a little bit what I'm doing now. But the other reason for plotting is for data communication. And this, in this case, I will want to do something, that would then be nice to show to other people, not just for myself. And these are actually two different uses, because for data exploration, often, I don't care so much that the plot looks a little bit ugly. It's fine to use the defaults of the software, but then for data communication, I'll want something a little bit nicer. So if it was just me for my own personal knowledge, I would stop here, but let's do a slightly nicer version where we build this slightly nicer plot. So we can do that. So we need to do one of these matplotlib things with matplotlib multiple axis things. Axis in one plot. No, it's not multiple axes, multiple panels. So it's these subplots. Okay, so I think we want this. Yeah, I think we want something like that. X, fig X. So we want two subplots, 2 by 2. But I think we can. We can specify the. Okay, well, we want two subplots, 2 by 2. We want them to share the x-axis, yes, true, and we want them to share the y-axis, true. And we want, not sure that this is the exact size, but let's say it's probably around, this is probably correct. So this is, oops, sorry, messed that one up. Yep, that's where I want it. So the contamination, it's here, right? So is that 1, 0? And okay, So I want that one to be vertical. How do I do that? So I want that to be vertical. How do I do that? I want that to be... Can I get a vertical? So one way, yeah, this is what I want. Yeah. Okay. Oh. Oh, sorry, that's the wrong one. Oh, there it is. Oh, sorry. Okay, yes, so that's there. Yep, okay. Kind of. Okay, I think this is actually what I wanted. Let's just, okay, I'm gonna okay, I know it looks very ugly right now, but I'm gonna, so if we do it the other way around, you could, okay, the other way around. So, okay, so that will be, okay, so let's see, this, so it's gonna be completeness here and contamination there. So I want the completeness histogram there, so that's 0, 0. Yep, there's the completeness histogram. I want the contamination histogram there. That's 1, 1. There's the contamination. And now I want the scatter plot. I think this is there, so it's 1, 0. Very nice. Okay. I want to remove that one, so that's axis 0, 1. Is that a set visible thing? Yep. Move. Okay, and now I think the big problem here is that for some reason, so this is the contamination. I just wanted to go from 50 to 100. And that goes from 0 to 10. Right. Sorry, there's something here that something I don't understand. What? Okay. All right. I'm gonna restart everything. Let's look at what we did. So, I think we're almost there. Okay, so we do this. Okay, I'm gonna remove the stuff. Okay, so this is what, okay, so I think this is the right idea. Share. Okay. Sorry, okay. No, this is the, okay, so I get the, okay, because let's see, it's the, oh, sorry, yeah, this is, I messed this one up. So I have, oh, okay, I messed up there the indices. Yeah, sorry, I messed the indices. Okay, this is the problem. Okay. Okay, all right, so that would be, oh, sorry, not yet. What if here's two? How did I? You have, so that's the, why is this not showing? Not, I actually expected that this would show up there. Oh, is it just the, oh, okay, you know what? Oh, okay, I know what's wrong. This should not be sharing the x-axis with that one. Ah, I see. Fake plot subplots. Yeah, I think share x should have been row and column. Okay. Okay. Okay, right. So, okay, so this is, so what happened here was that, yeah, so what happened here was that they were sharing the x-axis. So when I set the x, so the x-axis was messed up. So when it was setting the x-axis for the histogram, they should, yes, this is what we wanted. They really should be independent. The x-axis for the histograms, right? This is, so this here should go from, oh, actually, did I mess this up again? Share x across, okay, no, it should, okay, yes. Right, but yeah, so it should, so yeah, so this two, this top one and this bottom one, they should share the axis. the x-axis. And this one should share the y-axis. Sorry, yes, that's correct. But this one, the histogram shouldn't be sharing any axis. Right? When I mean sharing axis, I mean that, you know, this 4 here is the, you know, so where's my, this 4 here applies to the whole thing, not just to, so this 4 here applies to both this plot and this plot there. Okay, all right, this is what I wanted. Okay, so perfect. Okay, I'm gonna do a bit of cleaning up. I think we don't want this one there. Oh, sorry, we don't want both plots. We only want one of them. And actually, I am gonna save the figures. So, yeah, I could also have integrated this into the other script. You know, today for the partly for the in interest of time, I am not going to do that. So we could have made this a single script, but let's just get. Okay, so that's just check that things work. Yeah, it works pretty well. Pretty well. I mean, there's a so if I remove this, does it look much worse? So you can see that here there's this yeah, okay, yeah, that's better. Yeah, you could see before, you know, there was this very strict line at 100, and the markers are actually a little bit bigger than 100, so that was not great. So that gave this weird visual effect I didn't like. Now let's just do a little bit of, a little tiny bit of marker size, tiny bit of visual, making them a little bit small. Okay, maybe it's not marker size. Maybe it's, I think MS works. X is, yeah, let's set labels. X label completeness, and actually Y label contamination. MS. It's not MS. What's the? Is it just size? Scatter. Or just S? I think it's just S. Good. If okay. Yep. Bit nicer. Okay, maybe this. Some of this is frankly just trial and error. Yeah, but it looks okay to me now. I think it's a pretty. Okay, I'm gonna add to Git. Okay. What am I doing here? Maybe just now I'm just. So previously I was saving a PDF and the PNG. Let's just save the PNG. The reason being that it's actually, let's save it in SVG format and adjust the README to save in SVG. I'm just cleaning up. And I'm gonna add it to create completeness. Okay. Panel. And now actually, so normally I would not care so much about this, but just for the purposes of, yeah, you can see that, yeah, so it's on GitHub, it will also show, this, I think this is a pretty decent figure for, potentially for, if it was a real paper, we would still make it a little bit nicer. Right now, we're just, we're just making everything blue. We're not taking advantage of the possibility of adding color to add meaning. But for now, for about half an hour of work, it's not bad. I think it's also nice. I think so if you look at the scatter plot, it's also nice because there's a lot of dots here. And I think that having the additional information from the histograms, completes your mental model quite well, because there's a lot of dots there. And actually, so most of the dots are there in this small area, which is good. So these are the high quality genomes. So normally, so there's actually a standard for this. So, 90% complete, 5% contamination. This is high quality. We actually also want a couple of other things, namely seeing certain types of genes, but just from, so this box here is where we want to see things, and we see that there's most of our dots are there, and then there's a smattering of dots over there. Right, so I think I kind of ran a little bit out of time. I wanted to look a little bit at other things that we also get. In particular, I wanted to look at genome size, which is also a nice thing to look at. Let's convert it to megabase pairs. So genome size is also nice. So you can see there's one genome has 5 megabase pairs. It's probably very complete. 2.6. This is pretty complete, but actually some bacteria are even smaller than 2.6 megabase pairs. So some bacteria are even smaller than 1 megabase pair, but those are those are exceptions, or those are not exceptions. It's more of a continuum. Those are the tail end of the curve. So I think this still represents that most of these are still a little bit incomplete, but not so bad actually. So I still wanted to look and the total contigs. So maybe we will look a little bit more at this next week, and also, so this is the number of contigs. So these are long read sequenced. So this is why they tend to be so long, each individual contig. So this is the number of contigs, and you can see that there's a relatively large variation, but even at the top, there's the most fragmented genome has 64 contigs. but the median is 6, and the 25th percentile is 1. So that's a single contig genome. So maybe next week we will look a little bit more at this, and also trying to look not just at the overall, but maybe we can pick up this plot and add an extra layer of information with the with the number of contigs or the length by using the color, something like that we can explore a little bit. So I mean, so this was the motivating example, is looking at these genome qualities, but obviously a lot of these are sort of data science, sort of general purpose. You have a table of elements, and so here basically this is one way to have a plot one against the other, where you have both the scatter plot and then the histograms on the margins. Sorry, the histograms on the margins happens to be related to completeness and contamination, but this could be any other type of table. And then, as I said, we're not making use of the color dimension. We might try to do that next week, and trying to add the number of contigs, or the completeness, or the genome size, or I'm not sure we want. Maybe we want to look at some of these just to just to be sure we really understand what CheckM2 is giving us, but those would probably be the important ones. All right. Oh, there's something. Okay, this question. I'm just going to read. So, do you look at the histogram of length as well as mag QC? Generally, only look at completeness and contamination. That's it. So generally speaking, the automated programs will use length. We use completeness and contamination. So that's what they So even here, so we use them as a filter, so as a strict filter. So we just want, and then as I said, there's also these standards about what counts as a high quality mag, and that actually looks at completeness, contamination. It also looks at something we're not looking at today, which is the presence of certain kinds of genes. So you want to have the ribosomal protein genes, you want to have the ribosomal non-code RNA genes, and you want to have the tRNAs in there to have really a very high quality genome. And then we actually also then look at total. So looking at the total length, the total completeness, the total number of contigs, which is, so ideally you have a single contig, right? Ideally you have a single circular contig. So the more contigs you have, the worse. Means, for the same number of base pairs, you want to have it as one contig rather than multiple ones, right? So the more fragmented your genome is, the worse that is. So those things you should be looking at them. You should be looking at all of them, but completeness and contamination, or completeness and contamination as estimated by CheckM2, is normally the first thing that people look. Is normally the first thing that people also use to filter. So in this case, anything that CheckM2 didn't say was at least 50% complete, we don't even look any further. So that's normally the first check, that the one that's almost automated. We don't even look too much more. But then the other parameters are also important. I also always like to have a little bit of a vibe check, if you want to, just sort of look at what the data looks like, see if there's any big outliers, the most extreme cases, are they, they tend to be where where you have some sort of artifact there somehow, or where you have some biology that you're not understanding. So both of those are good things to understand better. Either your process is not working how you think it is, or there's some interesting biology that's not what you think it is. Either way, so I do like to look at these other things, but most of the automated processing will look at completeness and contamination. So this is so next week we will look at a few of the other things. Also, the other maybe point I'll make is that completeness and contamination they're very standard tools for this. So there's CheckM2 is maybe the most widely used. Some people still use the first version of CheckM. There's also a couple of other tools, but these are very you run a tool, it gives you that for estimating things like the presence of tRNAs, or there's maybe less consensus on exactly how you do that, and there's a couple of tools and they will give you slightly different answers. So maybe that's also something to take into account. They are also a bit less easy to run. So maybe that's also partly why people focus on completeness and contamination. Maybe also the last thing there is, so Illumina genomes, generally speaking, are, so if you get the MAC from Illumina sequencing, short read sequencing, they tend to be much more fragmented as well. And often they are specifically lacking ribosomal genes, both the rRNA and the protein ones, just because ribosomal genes tend to be repetitive because they're conserved across the sample. So they're not repetitive in themselves, but they are conserved across the sample, etc. Because different strains will have, similar but not identical versions, and this is what confuses the assemblies when you have things that are similar but not identical, because then it doesn't know how to put them together. So that was a very long-winded answer to say that yes, you start with completeness and contamination. That's a very well understood. People agree. There's a pretty broad agreement on how you should handle those. And then there's less agreement on how to handle the other metrics. But I would also, you should also be looking at your data, at least in some sort of exploratory way, to be comfortable with what you're doing. No. All right. So let me just double check. Okay, I pushed everything to GitHub. Everything's there. I will update on the Dropbox as well after wrapping up. Lawrence, you want to say a few things to take us out? I'm going to stop sharing. Thank you. All right, thank you.